7 discoveries show how lowering the APOE4 gene could actually protect your brain. I'm Dr. Kevin Tran, and if you carry APOE4 like I do, you probably heard the same warnings over and over again. It's very high risk, there's no cure for Alzheimer's, try to stay healthy, do some sports and so on, which is true. But behind the headlines, scientists are quietly rewriting that narrative. So this video breaks down 7 major insights from the Alzheimer's Association International Conference on APOE and Lipid Biology that happened in March 2025. So not that long ago. It's extremely new research, cutting edge, and you need to know that. You will hear about some of the world's top researchers, and most importantly, will translate these findings into clear, actionable strategies for people like us. From gene silencing to microglial precision therapies, these breakthroughs aren't decades away. They're actually happening right now, and they could change how we prevent, delay, or even beat Alzheimer's if you carry APOE4. Let's dive in. Here's one of the biggest unanswered questions in Alzheimer's research. Is APOE4 dangerous because it's doing something wrong, or it's dangerous because it's not doing something enough? And it's the difference between a criminal causing damage and a firefighter failing to show up. And that difference matters. If APOE4 is toxic, we want to shut it down, but if it's just ineffective, blocking it further could make things way worse.
We want more or less APOE. And directly related to that, the important question here is whether APOE4 represents a gain of function or loss of function.
So how do we answer that? We look at how much APOE protein your body actually makes. Across genotypes, here's the pattern. APOE2 makes the most. APOE3 is in the middle. APOE4 makes the least. And here's the kicker. The lower your APOE levels in the blood, the higher your Alzheimer's risk.
Plasma APOE levels follow this pattern of higher APOE2 compared to APOE3 compared to APOE4 levels. Inversely, we can really see the opposite pattern for Alzheimer's disease risk.
So at first glance, it seems like more APOE equals more protection. It's not true in the brain. That's where the story flips, because blood levels and brain levels don't always move in sync, and their roles might be totally different.
So we can ask whether these represent actually side effects of the APOE genotype, or whether this impact on APOE levels has a direct causal link to Alzheimer's disease. Which raises the question, should we decrease or increase APOE4 levels and even APOE3 or APOE2 levels?
That's why, by the way, your blood test might be a very imprecise proxy into what happens in your brain. So you can have perfect blood biomarkers and still have some issues in your brain. So be careful about imprecise proxies. This is where the science pivots from a numbers game to a function game. Let's step back and decode this. APOE, short for apolipoprotein E, is like a molecular delivery truck or a taxi. Its main job is to move cholesterol and fats where they're needed. In the body, this helps clean up after a big meal. But in the brain, it's about maintaining myelin, repairing neurons, and keeping inflammation in check. So here's where it gets interesting. In the brain, APOE isn't made by neurons. It's made mostly by astrocytes. Those are the brain's maintenance crew. And microglia. Those are the brain's immune defenders. Now, APOE4 does 2 things. 1, it's produced in smaller amounts. 2, the protein it makes is misshapen and unstable. So APOE4 may be doing less and doing it worse. Think of it like this. APOE2 is a very high efficiency logistic team. APOE3 is, let's say, your standard Amazon or UPS van. APOE4 is an overworked delivery driver with a broken GPS, he's lazy, and it's a much smaller fleet of drivers. So that raises the central question. Is Alzheimer's driven by the lack of deliveries or by the wrong deliveries going to the wrong places? And that's critical, right? Because depending on that answer, our interventions will look absolutely different. If it's a loss of function, we might boost APOE4 production or stabilize the protein. But if it's a gain of function, a gain of toxic function, we may want to silence the gene entirely. Right now, researchers are running experiments on both ends. Some are developing antisense therapies to reduce APOE4 levels, while others are engineering corrected version of the gene or protein. And the blood-brain disconnect, that's more than a curiosity, it's a clue. It tells us that what protects the heart may not protect the brain, especially in APOE4 carriers. So let's look at what happens when you actually try to lower APOE. Researchers turned to mouse models, and what they found wasn't subtle. Whether you reduce APOE using gene editing, synthetic antibodies, or antisense oligonucleotide, you constantly see the same outcome. Less amyloid plaque, less inflammation, less neuron damage. In other words, slowing the root processes of Alzheimer's.
Reducing both APOE3 and APOE4 appears to be protective against Alzheimer's disease pathology and neurodegeneration.
So even a partial reduction, just cutting APOE levels by half, still gives strong protective effects.
When we administer antisense oligonucleotides or ASOs targeting APOE, we have shown studies reporting that targeting APOE3 or APOE4, achieving 50% reduction in the APOE levels appears to be protective.
That's not a small tweak. That's rewriting the trajectory of the disease before it takes hold. Okay, let's break this down. What are ASOs? ASOs are short for antisense oligonucleotide, are like genetic post-it notes. They stick to a piece of RNA and tell the cell, don't make this protein. In this case, the protein is APOE, specifically the APOE4 variant that's linked to higher Alzheimer's risk. And here's the analogy. Imagine your brain is a factory. APOE4 is a malfunctioning machine. Maybe it's overheating, mislabeling lab packages, and even leaking toxic waste onto the floor. ASOs don't destroy the machine. They quietly unplug it before it causes more damage. And when scientists did this in Alzheimer's-prone mice, something amazing happened. Plaques stopped building up, neuroinflammation dropped, and cognitive decline slowed or reversed. So even targeting APOE3, which is considered the neutral version, produced benefit. That tells us something big. It's not just about a toxic gain of function or low expression. It may be that too much APOE activity in general is harmful in the context of disease, especially if that activity comes from cells like microglia, which can act like overzealous cleanup crew, triggering inflammation when they are supposed to help. By the way, I have another video on microglia where we focus on 11 insights and more about them. You should definitely check that out. So that's why lowering APOE even slightly can tilt the balance towards brain protection. Of course, mice aren't humans, and this is the final word. But these results are strong enough that ASOs therapies targeting APOE4 are already moving towards human clinical trials. And for carriers of APOE4, it opens up a new possibility. You don't always have to fight harder. Sometimes you just need to quiet, the right signal. All right, we've seen that reducing APOE helps in mice, but mice aren't people. So the real question is, what happens when humans are born without APOE? Turns out we found a case that rewrites the rule.
An individual that carries 2 APOE loss of function, there is only one such individual that has been reported in the literature. This individual showed no evidence of vascular disease, and interestingly to the discussion today, was negative for A-beta and tau in CSF, normal on brain MRI, and grossly normal on cognitive status.
This is stunning, right? This woman had both copies of her APOE gene turned off, no backup version, no functioning APOE protein, and yet she had no cognitive decline, no vascular diseases, and normal brain scans well into late adulthood. That's like discovering someone who never changed their car oil and their engine is still like purring at 70.
So at least some things that are safe to conclude here is that APOE in the brain might not be that essential, and the therapeutic strategies that reduce APOE levels will likely not adversely affect cognition.
Let's unpack that. APOE has always been seen as essential, especially in the brain. It's the main carrier of cholesterol, and it plays a big role in synaptic repair, cleaning debris, and maintaining neurons. So the idea of completely turning it off used to be unthinkable. But this case changes that. This woman had biallelic loss of function mutations, which means both versions of her APOE gene were essentially broken. Think of it like a 2-lane bridge where both lanes collapsed, and yet traffic is still flowing perfectly on alternate routes. Her brain found workarounds, likely through other apolipoproteins like APOE1 or APOJ, or alternative cholesterol pathways. What's even more important, she wasn't just surviving, she was thriving. No signs of dementia, no major heart disease, and no obvious downsides to having no APOE. That gives researchers confidence to pursue APOE-lowering therapies more aggressively, because the big fear has always been, if we lower APOE, will we cause side effects like memory loss or vascular problems? So this case says, maybe not. Now, of course, it's just one person, n equal 1, it's a rare exception, it's not the rule, a lot more research is needed, but sometimes it only takes one outlier to change the entire paradigm. So for APOE4 carriers like us, it's a powerful proof of concept. Reducing APOE doesn't necessarily mean losing brain function. In fact, it may be a step towards preserving it. All right, now let's look at something less drastic. What happens if you partially miss APOE? Not a full knockout, just one copy of the gene code. Thanks to advance in whole genome sequencing, scientists have now identified people walking around with exactly that. One working copy, one genetic blank. And the results are eye-opening.
One individual, which was an E3/4 subject, had the knockout for APOE together with APOE3, leaving the individual to be effectively an APOE4 carrier, which is very interesting to compare to an APOE4 full individual. This individual developed Alzheimer's disease 5 years later than the typical APOE4 carrier, which is, in my knowledge, quite compelling.
That's huge. This person had one high-risk APOE4 allele, but their other copy was totally non-functional. Yet they got Alzheimer's 5 years later than people with two working APOE4 copies.
Another very compelling individual, which is effectively an APOE3 carrier, was still cognitively normal at 90 years of age. And when we look at this individual here in the red dot, when we look at pathology for Alzheimer's disease, cerebral amyloidopathy, and so forth, compared to APOE4 carriers, they really are an outlier having low pathology.
That's not just delay, that's resistance. Imagine APOE as a faucet, controlling how much cholesterol handling and immune signaling gets poured into the brain. These partial knockouts suggest that turning that faucet down, even halfway, can slow the damage. Let's break this down. Your APOE genotype is normally a pair. You inherit one allele from each parent. So most people have combinations like 3-3, 2-3, 3-4, or in high-risk cases, 4-4. But in these rare partial knockouts, one of those alleles is a null variant. It doesn't produce any function in APOE protein. So someone with APOE4, nothing, isn't getting the full dose of that risky variant. They're getting half. And the effects, it's delayed onset, low amyloid, better cognitive function, even at age 90. It's a bit like having a faulty engine, but one of the cylinders isn't firing, so it can't do as much damage. Importantly, these aren't engineered experiments. These are natural experiments, real-world evidence, real people walking around with less APOE and doing surprisingly well. And that reinforces what we saw in mice and ASO studies. Less APOE doesn't mean worse outcomes. In fact, it might improve them. For APOE4 carriers, this adds to the growing evidence that you don't need to silence both copies to see benefits. Even reducing the harmful signals pathway can buy you time, maybe years or decades, before pathology starts to build. And when you're talking about Alzheimer's, every year matters, right? All right, a quick pause here. If this kind of practical science helps you rethink your own Alzheimer's risk, gives you hope, tap subscribe below. These videos break down the most important insights from the latest research in a way that is clear, hopeful, and real. And hit the bell button so you don't miss any of my future videos. We're moving on to the fifth insight. If we zoom out from this rare mutation and we look at large population data now, if you measure APOE levels in the cerebrospinal fluid, that's basically the clear liquid bathing your brain and spinal cord, what do you find? You find a clear trend. More APOE equals more Alzheimer's risk.
Actually, higher levels of APOE CSF protein levels are implicated with higher Alzheimer's disease risk, which would be concordant with us wanting to lower APOE levels.
Let that sink in. We often assume more of a protein means better function, but here the opposite is true. In the brain, not the blood, excess APOE may be fueling the disease, not fighting it. And this matters especially for APOE4 carriers because your brain is already dealing with broken cholesterol transport, heightened inflammation, and hyperactive microglia. And more APOE on top of that, well, it's like pouring gasoline on a smoldering fire. Let's just unpack what this all means. So your brain floats in CSF, your cerebrospinal fluid. That's, again, the clear liquid that removes waste and delivers nutrients to your brain. Scientists use CSF to measure early signs of brain changes years before symptoms start. And by the way, that is way more accurate than any blood biomarkers. The issue is super painful to tap into your cerebrospinal fluid. So in the study, they measured APOE protein levels in the CSF across a large group of participants, and they saw a striking trend. As CSF APOE levels went up, so did the risk of Alzheimer's, and this held true even after adjusting for age, sex, and APOE genotype. So they were trying to remove all the confounding factors. So we're not just seeing correlation by genotype, we're seeing direct evidence that higher APOE inside the brain may itself be harmful. Why? Because APOE4 is sticky. It tends to bind to other proteins like amyloid and tau, making them even harder to clear. It can also overstimulate immune cells and microglia, triggering unnecessary inflammation. Again, we have a video on microglia by itself, and I suggest you actually watch it. And also, APOE4 is less efficient at repairing myelin, which means your neurons lose their insulation faster. So having more APOE4 in your CSF might not be a sign of health. It might be a warning light on the dashboard. This flips the old logic on its head. We used to think the problem was too little APOE, but in the brain, too much of the wrong kind, especially APOE4, can tip the scales towards disease. So this aligns with what we've seen across all the 5 insights we've seen in this video. Lowering APOE can reduce plaques and inflammation, partial knockouts delay Alzheimer's onset, even total loss of function may be surprisingly well tolerated. For APOE4 carriers, it's very empowering because it means that turning down the volume on your APOE4 genes might not just be safe, it might be one of the smartest things you can do to protect your brain. So we've talked in this video about lowering APOE across the brain, but what if we could go one step further and target the exact cell type causing the most trouble? That's exactly what this next study uncovered. Scientists found a genetic variant that lowers APOE levels, but only in microglia, the brain's immune cell. And the result? Less amyloid buildup in blood vessels and lower Alzheimer's risk.
A genetic variant was associated with reduced APOE levels specifically in microglia, and this variant is also associated with reduced cerebral amyloid angiopathy and Alzheimer's disease risk.
This is huge and tells us where APOE is produced matters just as much as how much it is produced. And it makes microglial APOE4 looks like a precision target, not a global villain. So why microglia? Think of microglia as the brain's janitor and security guards. They're constantly scanning for damage, debris, and intruders. But when they carry APOE4, they don't just clean up. They can become, let's say, trigger happy, super overreacting, inflaming nearby tissues, and even releasing harmful cytokines that damage neurons. Now here's the twist. In this study, the protective genetic variant didn't lower APOE in the whole brain. It only lowered it in microglia. And that was enough to see a real reduction in Alzheimer's risk and vascular amyloid pathology. That's like adjusting one machine in a giant factory, and suddenly the whole system runs smoother. This gives us a powerful clue. We don't need to silence APOE everywhere. We just need to quiet it in the right places, especially inside the immune cells that overproduce it. And for APOE4 carriers, that opens the door to so many targeted therapies. We're looking at ASOs or CRISPR edits that are specifically in microglia. You can look at biomarkers panel that detect inflammation linked to APOE activity, or precision medicine tools that tweak expression without a full knockout. So what's the bottom line of all of this? Well, after decades of debate, the evidence is converging. Lowering APOE, especially APOE4, isn't just safe, it might be one of the most effective strategies we have.
Multiple studies clearly support that lowering APOE4 should be the target for therapeutic approaches for APOE4 carriers, which I essentially agree with.
That's a bold statement, and it's backed by mouse studies, human case reports, partial knockouts, and population-wide biomarker data. It isn't just a hunch, it's a shift in the scientific consensus. And it opens the door to new generation of precision therapies. So again, ASOs to reduce APOE production, microglial targeting modulators, and even gene editing approaches to selectively quiet APOE4. We're not there yet, but clinical trials are already underway. So to summarize a little bit, for years, scientists were afraid to touch APOE. It's involved in so many processes, right? Lipid metabolism, synaptic repair, immune signaling, that any attempt to reduce it was seen as too risky, not worth it. But these studies flipped that fear on its head. Mice lived longer and performed better with less APOE. Humans missing one or even both copies showed delayed or no disease. CSF biomarker studies revealed that more APOE4 in the brain correlates with more damage, not less. So instead of APOE being a can't live without a gene, we're now asking, what if it's not essential at all, at least in adults? And more importantly for us, what if too much APOE4 is part of what's driving the disease? So if that is true, the best approach isn't to block amyloid. It's not to chase tau protein and so on, or to do guesswork with supplements. The best thing we can do is to go way upstream and just tune down APOE4 itself. That's why at the Phoenix, we follow these developments so closely. All right, we had tons of insights in video. Let's recap everything. Insight number one, we started by asking, is APOE4 toxic or just missing something? Is it a gain of toxic function or is it a loss of function? Turns out it's a gain of toxic function and you want to lower it. Insight number two, build upon that. In mouse models, reducing APOE, whether by gene editing or antisense therapy, led to less amyloid, less inflammation, and slower brain degeneration. Then, not only looking at mice, we looked at humans. One woman with 0 working APOE genes was cognitively normal, suggesting that the brain can adapt and can use other pathways to transport cholesterol and fat. And it means that not having any APOE genes is okay after all. Insight number 4, we looked at knockouts. What if we partially knock out APOE? So people with one broken APOE copy. So those had a delayed onset or no Alzheimer's at all. So overall, the insight is that less APOE you have, more protection you have. Insight number 5 looked at higher APOE levels in brain CSF. Those are linked to higher Alzheimer's risk in the brain. More APOE isn't better, especially for APOE4. Insight number 6, we look at genetic variant that lowers APOE specifically in the microglia, so you don't need to touch APOE anywhere else. You only look at APOE in the microglia, and that was protective. That shows us exactly which cell types to target. So you see that slowly we're actually building the precision medicine and targeting in a way that is more and more precise. Insight number 7, across all this research that summarizes it well, one thing is clear: lowering APOE4 is not dangerous, and it might be one of our most promising strategies. Cool, since you watched until the end of this video, I am betting you're an APOE4 carrier. So if you want to beat the odds and defeat Alzheimer's, check out the Phoenix community. The link to apply to join us is in the description. I review all applications personally to make sure that everyone is a great fit. So if you're coming from YouTube, just mention it. I'll make sure that you get some extra love. Thank you for watching, and I'll see you next time. Bye.